Hello everyone, welcome to our new video on preclinical pharmacology. In this video, we will discuss about the enzyme myeloperoxidase, which is a versatile enzyme in health and diseases. Let's see the introduction of myeloperoxidase. Myeloperoxidase is an enzyme and a type of protein that speed up the chemical reaction. It is generally found in the granules of neutrophil, which is a type of white blood cell. Myeloperoxidase is located at chromosome number 17 in a human and at chromosome number 11 in a mice. Approximate molecular weight of myeloperoxidase is a 150 kilodalton. Let's see the structure of myeloperoxidase. Myeloperoxidase is a composed of a heme group and the protein structure. Heme is a prosthetic group and which is responsible for the enzyme's catalytic activity, binding activity, and it is also helped to activate the oxygen molecule. Protein part of the myeloperoxidase is composed of a two subunit, that is a heavy chain and light chain, which is linked together by a disulfide bond. Let's see the subunit and assembly of myeloperoxidase. The heavy chain of myeloperoxidase contains the heme group and the active site. The light chain is involved in the stabilizing the structure and regulate the activity of the enzyme. The heavy and light chain assemble to form a dimer, and dimer is an active form of myeloperoxidase. Let's see the discovery of an myeloperoxidase. Myeloperoxidase was first discovered in 1917 by the scientist Alexander K. Ward. In 1950, further research leads to the characterization and isolation of the enzyme which was named as a myeloperoxidase. On a present day, ongoing research is continued to uncover the complex mechanism and the function of myeloperoxidase in health. Let's see the myeloperoxidase derived oxidant. First is a hypochlorous acid, that is a HOCl, which kills the pathogen and damages the bacterial membrane. Second is a hydroxyl radical. Hydroxyl radical is a highly reactive species which can damage both the pathogen and a host cell. Third is a singlet oxygen that damages bacterial DNA. Now, the biological function of myeloperoxidase. Myeloperoxidase produces a hypochlorous acid, which is highly toxic to the bacteria and pathogen. Then the inflammation. Myeloperoxidase contribute the inflammatory response by generating the reactive oxygen species and activating the inflammatory pathway. Myeloperoxidase also play a role in a tissue repair by promoting the clearance of damaged cell and debris. Myeloperoxidase also has an antioxidant property which helps to protect the cell from damage caused by the reactive oxygen species. Let's see the role of myeloperoxidase in the immune system. Neutrophils act as a first line of defense against the invading pathogen that include bacteria, virus, and fungi. By the process of phagocytosis, neutrophils engulf and destroy the pathogens. Myeloperoxidase is a key component that helps to fight against infection. Let's see the role of myeloperoxidase in regulation of inflammation. Myeloperoxidase contribute to the inflammatory response, which is a complex process and help a body to fight against infection. Myeloperoxidase also help to activate the neutrophil, which release the inflammatory mediators. Then the myeloperoxidase also play an important role in resolving the inflammation once the infection is under control. Mechanism of a antioxidant action of myeloperoxidase. First mechanism is ROS scavenging. Myeloperoxidase directly scavenge the reactive oxygen species and neutralize their damaging effect. Second mechanism is antioxidant enzyme activation. Myeloperoxidase can indirectly activate other antioxidant enzyme and enhance the cell defense system. Third mechanism is a redox balance restoration. Myeloperoxidase contribute to restoring the redox balance within the cell, which help to prevent oxidative stress-induced damage. Let's see the beneficial and detrimental effect of myeloperoxidase. Beneficial effect. The antimicrobial activity of a myeloperoxidase is essential for protecting the body from the infection, especially those caused by the bacteria and fungi. Detrimental effect of myeloperoxidase is nothing but the harmful effect. Overproduction of a ROS by myeloperoxidase can cause a condition such as atherosclerosis, rheumatoid arthritis, and the cancer. Let's see the clinical implication of myeloperoxidase dysfunction. First condition is atherosclerosis. In atherosclerosis, the increased ROS production by myeloperoxidase contribute to the development of atherosclerotic plaque. In rheumatoid arthritis, elevated level of myeloperoxidase is associated with the inflammation and joint damage. Myeloperoxidase also play a role in a cancer progression by promoting the angiogenesis and tumor cell proliferation. These are the tests which is used to measure the level of myeloperoxidase. First is the ELISA, that is enzyme-linked immunosorbent assay. ELISA measures the amount of myeloperoxidase protein in a blood sample. Second method is the immunological technique. This technique uses antibodies to specifically detect and quantify the level of myeloperoxidase. Interpretation of a test result of myeloperoxidase. Normal range of myeloperoxidase may vary depending on the laboratory and the method used for the testing. Elevated level of myeloperoxidase may indicate the inflammation, infection, and other conditions. Low level of myeloperoxidase may be associated with the impaired human function or the certain genetic condition. Future direction in a myeloperoxidase research. First is a therapeutic target, exploring a myeloperoxidase inhibitor as a potential treatment for the inflammatory disease and cancer. Second is a diagnostic marker, investigating myeloperoxidase level as a potential biomarker for the disease progression and risk assessment. Third is the immune modulation, understanding the interplay between the myeloperoxidase and other immune components to develop the new strategies for immune modulation. Thank you.